Hi, everyone. We're very lucky to have April Khadeni as our speaker today. So April is a Canada Research Chair in AI for Medical Imaging and Associate Professor of Biomedical Engineering at TMU and a faculty affiliate at Vector Institute. April's research focuses on the design, development, and deployment of AI algorithms for medical imaging. Her research is funded by the Canada Research Chair Program, CIHR, NSERC, Ontario Government, Alzheimer's Society, and Canadian Cancer Society, and MITAX. She has a PhD in electrical engineering from U of T and is a licensed professional engineer in Ontario and an IEEE senior member. And today she's going to tell us about leveraging AI for medical imaging.
Thank you very much. Hi, everybody. Welcome. Thank you very much for coming to my talk today. And the title of it's Leveraging AI for Medical Imaging, and I will discuss various aspects of design, development, and deployment of AI for medical imaging. So if you consider this, okay, there's a bit of a lag here. Okay, there we go. So if you consider this picture, it's the anatomy lesson of 1632 by Rembrandt. It's a bunch of physicians. And in order to learn about anatomy, at that point in time, they actually had to cut the patient physically open. If you contrast that to today, 2024, we have many more tests available. We have imaging tests, signals, genomics, phenomics. Not only is there more tests available, but these are largely captured and stored in a digital format. So what this presents us with is a unique opportunity to utilize and leverage high-performance computing computers and artificial intelligence algorithms to gain unique healthcare insights, to go towards a more personalized medicine paradigm. Out of this kind of healthcare AI or medical AI, medical imaging is a critical piece of the puzzle. Not only is it 80% of all healthcare data in hospitals and institutions, but it also shows you the way that the disease has manifested in the patient today. And as a result, it's a critical piece in diagnosing and providing treatment options for patients. However, as with any type of manual analysis, visual interpretation of images is subjective, laborious, and error-prone. And what this affects means that perhaps you could have a radiologist, let's say you have two radiologists reading the same image, they might come up with a different answer, and this could lead to the patient being assigned different therapies. So this affects quality of care. So we can utilize AI for more objective and efficient and new insights. So that's what we do at my lab, the Image Analysis and Medicine Lab. We're extracting insights from medical images, and we're using AI to analyze, summarize, quantify, integrate, and model medical imaging data in a clinically meaningful way. One of the high-value applications is this patient stratification and using these biomarkers in order to determine whom to treat and when. And this drastically improves quality of care. For the last several years, we've really focused on workflow augmentation. So providing clinicians, such as radiologists and pathologists, with the necessary software tools for them to get measurements from the imaging data. And so here's an example. Here you can see the algorithm results, and then you would have a series of metrics that were derived from the images, and these are used to guide therapy or therapeutic decisions. And so using AI for workflow augmentation improves diagnostic accuracy, turnaround time, and quality of care. So in my lab, we focus on the two major medical imaging modalities. One is radiology AI, which looks at gross or macro anatomy and pathology. And then on the other side of the color images is digital pathology. And this is looking at tissue and cells under magnification. And so today I'll kind of walk you through some of the applications that we've done. We've done the design, the validation, and we've started to think about the clinical considerations for both radiology and pathology. On the radiology side, we have more than a decade of experience, particularly in neuroimaging AI, and that goes from dementia, Alzheimer's disease, vascular disease, and also brain cancer. And the last little while, we've also focused on pediatrics. So I'll talk a little bit about some of the work we did in these areas. So like I was saying, for the last 10 years, we focused on neuroimaging analysis. We've developed complete pipelines from pre-processing to all kinds of object detection to biomarker extraction. And we've developed novel biomarker systems for neuroimaging. We focused on dementia and cognitive impairment for a long time, but we've started to extend it to cerebral vascular disease as well as pediatrics and brain tumors. Within this pipeline, and I'll talk about it a little bit, we have patent pending technologies that has actually enabled us to do wide-scale analysis of medical images. And we were actively processing 600,000 MRI robustly and reliably over 100 centers. But this is recently, that number has been increased to about 4.5 million data sets from 60,000 patients. And that's going to keep us busy for the next couple of years. But we've been actively processing the 600,000 images for several years. And from these systems, we've actually come up with new biomarker systems, and I'll talk a little bit about those today. And this is kind of gives you an outline of some of the deep learning-based segmentation tools that we've looked at, from pre-processing to brain extraction to white matter hyperintensity segmentation, which are these lesions in the brain. They're correlated to cognitive impairment as well as vascular disease. And I will also talk a little bit about ventricle segmentation. And these are the important markers that are usually used by clinicians to say, is there neurodegeneration happening and is it significant? So we've tried to automate this. So the first thing we developed was, so we focus on pre-processing. Generally, what you want to try and do is tidy up your data as much as possible before you create a model. So there's several pre-processing strategies available to neuroimaging AI. In this one, this is a patented technique where we normalize the intensity in MRI images. And so you can see the histogram. These are volume histograms of the intensities for all the different sites. Here you can see that they greatly vary. So for the same brain disease, same patient, if you go to different centers, your histogram is going to be vastly different. So this creates kind of a domain shift. And so one of the first things that we focused on as a lab was how to correct for that. And we came up with a novel intensity standardization method that we've applied widely to all our data sets. And we, as you can see here, all the peaks are aligned, and this corresponds to the brain tissue region. And so the brain tissue intensities are aligned. And this has enabled a lot of both our biomarker analysis as well as our deep learning based analysis. So another type of pre-processing technique is to reformat your data to have the resolution, the pixel resolution that is required by your network. In medical imaging, the slice thickness, so the Z direction, varies greatly. Sometimes you have 2 millimeters voxel resolution, sometimes 5. And so to normalize it, we proposed a novel network that we called MLP-SRGAN. And it basically uses MLP mixers instead of convolutional neural networks in order to upscale the image. So on the generator side, we have the MLP mixers, which we proposed for the first time for upscaling. And then we have a discriminator that basically ensures that the images that we've generated are realistic. The nice thing about this framework is that you can actually change the size of your filters, and you could actually upscale your image to any type of resolution that you want. So it's pretty flexible. And it might be hard to see here a little bit, but we've actually highlighted the regions that we've looked at. So at the top where it says original, that's the high resolution. And our MLP-SRGAN with one discriminator block is underneath. And what we found is that it preserves anatomical subtleties. It localizes tissue characteristics better. And actually in the paper, we've proposed a whole series of new metrics that are clinically inspired, that instead of just PSNR and SSIM, which is used widely in the computer vision literature. Another type of pre-processing algorithm we focused on is sometimes you might want to, for longitudinal analysis, you're going to want to align serial imaging, or you might want to align to an atlas. And so we propose an algorithm called FlowReg, which is a fast, deformable, unsupervised medical image registration technique using optical flow. So it's a deep learning registration technique. There's two components. It goes through the FlowReg A, which is the affine component. And we designed this network to predict the scale, translation, and rotation parameters. And actually, what we found, which was quite interesting, is that a lot of the heavy lifting was done by that network. And the fine tuning was done with the optical flow network, which actually tries to predict the voxel movement between fixed and moving images. So it's a vector field. And so you can see here that the deformation at the top, it did the fine kind of adjustments after. And at the top here is our fixed image. So that's the atlas we want to align to. The before is our moving image that we want to align to the atlas. These are all the comparative frameworks, and I'll just bring your attention to FlowReg, affine plus optical flow. So FlowReg, what we can see here is that both level with the eye all the way to the top has the best alignment. And we actually proposed a novel battery of validation metrics to actually, which is clinically inspired, to show that the registration get kind of a better understanding of where it works and where it fails. Okay, so now we've pre-processed our data. Now we can start to get ready to segment structures from our brains. So in 2017, we proposed a novel framework for automated brain extraction. It was based on classical image analysis, standardization with thresholding. And what you can see here is actually, these are the two outputs here. These are the ground truths. And you can see compared to what was the state of the art at that time, that our algorithm was performing better, and our quantitative metrics also showed that. So we're getting pretty high DSCs. One of the things we started noticing is, because we don't work with just normal control images, we work with pathological volumes. And these volumes sometimes, especially when there's like a lesion close to the periphery of the brain, it would cause this algorithm to fail. So we started to think about how can we do handcrafted feature design, which was like intensity, texture, intelligent sampling strategies, and a random forest classifier to perform the classification. So voxel-wise classification. And you can see after post-processing, that's the estimated brain mask. Another test that we did is we showed that if we don't use standardized data, that basically it fails across other scanners. So once we've standardized our data, we can, these are those typed box plots that don't have that variability. So during this time, after we just kind of wrapping up this, fortunately, deep learning started making its way to the medical imaging community, okay? It always takes a little bit longer to get to us, I find. It's sped up in the last few years. So what we did is we wanted to look at this task using UNets and UNet variants and see how it did. And so here are the results. I should mention that the total number of images that we use here is around 10,000, and from like 150 patients from 30 or 40 centers. So we always focus on multi-center validation and wide variability in our data sets. And you can see the results here. The first column is the original image. The red overlay is the ground truth. Then the two traditional methods, so the thresholding and the random forest classifier, and all the blue ones were the deep learning architectures. We found the deep learning architectures to be significantly more accurate, and we also found it to actually do very well in the upper and lower slices, as well as slices with pathology. So these were some of the problems that we had been dealing with for years that fortunately deep learning was solving for us. Another type of segmentation that we focused on, I actually focused on it for my PhD, so it's come a long way from completely unsupervised to now deep learning. Again, we validated on, and we trained and validated on about 12.5 images from 39 centers, so wide variability, 5 data sets. We even proposed, and it was kind of novel at the time, a 2 data split strategy. So we train on one data set and we test on all the other data sets, and we measure generalization performance. And so what we found was, again, that compared to the traditional methods, like you can see, these two are the traditional, and it's under segmentation and over segmentation. The UNet variants were doing an excellent job. And one of the things that we found it kind of interesting to see was when you look at the predictions from all the UNet variants, even though there's some differences in terms of like multi-resolutional or skip connections, for example, they looked very similar. So we also did a little sub-analysis where we overlaid all the predictions on top of each other, and we found that most of the predictions were all found the same regions, but the SCUNet and the multi-res UNet seemed to pick up some of that finer structures. So we really like the SCUNet actually as the top performer in this task. So the last several years, which actually it's from my PhD and now extending into my lab, we really focus on generalization and wide scale adoption of tools. So how do you make them robust and reliable? So in this case, we wanted to focus on a Siamese network with feature losses to force better generalization for WML segmentation. So on the generator side, we actually have just a modified UNet structure that generates predictions. And then we propose this novel discriminator, which is based on the Siamese network here, that tries to match the ground truth feature vectors to the predicted feature vectors. And what we found by doing this, and we did ablation studies without it, and we found that this discriminator actually helped to enforce better generalization to out of domain or out of distribution data. So you can see the top one is the held in data set. It's about, the Siamese loss is about 0.79. So it's about the same as like the top performer. And then on the held out of distribution data, it was significantly better. It performed better. Another work that we focused on is that in medical imaging, one of the main challenges we have is annotations. Like if you can see how detailed these annotations are, it can take up to half an hour to do one volume. So getting a lot of data can be tricky. And this is particularly a problem if, let's say, I train my algorithms on a Siemens scanner and then I want to go and test it on a GE scanner, for example. So what we tried to use was a cycle-GAN approach to generate data in the target domain. So we tried to take, for example, GE images, generate a Siemens one, and so on and so forth. And despite all the fine-tuning that we did, we optimized it, we even found ways to reduce memorization, we couldn't get the addition of these data to improve segmentation performance. That's why I'm like particularly excited about the next generation of Gen AI algorithms, because perhaps we can get more realistic or just better generated image data. I'll just also add one other thing that it's not here, but it's in the paper. We actually had an unconstrained image generation network, and I believe it was just removing the discriminator or something like that. And it generated images that were not realistic looking, but those images actually improved segmentation performance. Maybe it generates features that the classifier is looking for, I'm not sure, but it was very strange. They did not look like normal brains, and the lesions were everywhere, and those images actually helped. So somehow the features from those images, maybe not spatially dependent, improved it. And I'll just quickly talk about this. So we use our intensity standardization. We published this paper last year at Middle. And basically what we showed by standardizing the data and building a model from standardized data, both on in distribution and out of distribution data, you do much better, including on the small lesions, which are usually hard to detect. The last segmentation work I'll talk on the neurospace is this work we published last year at Middle as well. And what we did was we were trying to think about this translation aspect. So if I create, let's say I create a Vector Institute model, I have the scanner here, create a model, and I want to go to Sunnybrook or UHN or TMU, and I want to apply my model. There's about a 20% drop in performance, let's say. So how can we actually improve performance in target sites? So this one, we proposed domain adaptation using silver standard masks. So in the target sites, we would use unsupervised segmentation to automatically extract silver standards from the targets, use it to pre-train our network, which was fine-tuned with the gold standards. And we saw that this drastically improved performance in target sites. And this one, we had like many different data sets, many different centers, and we showed that it was a significant improvement. And if I can just show, maybe bring your attention to one of them. So you can see here, this is the ground truth here. And Brandon, you'll recognize that's Andre data set. And you can see that all the models missed it, except for the one that was fine-tuned using the target site. So this improves robustness. So we have a whole series of segmentation tools that we've validated in multi-center data over a number of years. And now we've started to look at the clinical application of these tools. So, okay, we've segmented stuff. So what? What are we going to do with it now? Let's start to extract biomarkers from them. So one of the most intuitive biomarkers is just volume. So if you've segmented something, you can get the voxel dimensions and get the volume from the number of pixels. And so we looked at white matter lesion volume and total brain volume in over 4,500 patients. And we were able to look over cardiovascular diseases, MCI, AD, which is mild cognitive impairment, and Alzheimer's disease, and another data set that also has similar characteristics. The interesting thing about this work is that we were able to actually do longitudinal change measurement, and that's extremely tricky because sometimes the change is smaller than the noise. So your tools have to be very robust in order to elucidate those fine changes. And we did that. So using the last year or so, we've enhanced our biomarker pipeline. We have regional metrics. We have all different types based on the white matter, penumbra, and different things. And we've published a number of clinical papers showing the clinical validity of the biomarker frameworks. In this paper here, we published in the American Journal of Neuroradiology. What we do is you take a whole bunch of normal brains, extract features, develop a model, a normative model to predict brain age. So if you have normal aging, your brain tissue should be healthy, and you should be able to predict the chronological age. But then when you run that normative model on people with Alzheimer's disease or mild cognitive impairment, you start to notice that the difference between the normative is getting bigger. So this is called the brain gap estimation. And the wider this brain gap estimation means that your brain is less similar to the normals. So you can see that how from like increasing the disease severity that this brain age gap estimate is getting wider. And so basically you can use it as an automatic triage or flagging tool to actually identify accelerated aging. And in this work, we use similar biomarker systems with also, I believe this was a random forest classifier. So we've extracted everything. And what we found is that we were able to predict which subjects are going to convert to Alzheimer's disease up to 3-4 years earlier. This is super important for a number of reasons. One, once you've accumulated brain damage, it's too late. You can't really restore neurons. So you want to always identify disease earlier. So that's the first thing. The second thing is that we are identifying which patients will convert to Alzheimer's, which means you could isolate or these subjects for basically clinical trials. The last work I want to highlight is basically we looked at Alzheimer's disease versus vascular disease. We were able to find a whole bunch of interesting relationships. I just want to bring your attention towards the end that what we were able to do was find this novel kind of clustering strategy that lets us actually threshold between disease groups. And this is the step right before you can use it for clinical translation, because I'm now able to use the measurements I'm extracting from images to put patients into subgroups. So there's that. And this work we are just about to submit. So using all these biomarker frameworks and segmentation and everything, we're starting to come up with biomarker frameworks and biomarker signatures of different patients that we can use to stratify people into homogeneous subgroups. The last neural application I will talk about is for pediatric neural development. This was published in American Journal of Neuroradiology, and follow-up will be coming in the Canadian Journal of Radiology. And basically, we had around 500 children from ages 0 to 20. We've extracted all these measurements from their brain, and we've developed normative neural development curves. So now if somebody has neural development issues or some kind of disease, you can compare it to the normative curves and find who's abnormal or developing abnormally. And that's what we kind of showed with this proof of concept here. So we've actually extracted measurements from a tumor volume, and we can see that actually even the normal appearing white matter has some alterations. Okay, so that I'm going to switch gears a little bit. So we were talking about radiology and neuroimaging. The next step, we're trying to actually translate some of these tools and work with a local hospital to see how we could actually implement these biomarkers. On the other side of my research, I focus on digital pathology, and digital pathology is a relatively new imaging modality that has been digitized. Whole slide scanners only have been FDA cleared in 2017. These images have unique challenges in that they're about 100,000 and 100,000 in pixels for one slide. So like a breast tumor could have, let's say, 10 slides. So you can think 100,000 by 100,000 times 10. It's a massive data processing challenge. And we're developing apps for this modality as well. So I'll kind of skip past this one. So a few years ago, we focused on Ki-67, which is a biomarker that indicates the aggressiveness of a tumor. And what pathologists actually do is they count the number of brown cells to the number of blue cells, okay? And so they used to actually have a bell that they used to ding, and that's how they would do the counting. And so we've implemented a deep learning-based system to automatically detect these nuclei and provide the measurements. And as you can see here, we have 4 or 5 data sets. So it's multi-center, multi-resolution. And what we found is that our deep learning architecture, which we called PyNet, did a great job. It worked on multiple types of modalities. So we have the patches. You can see here the greens are the blue cells, the browns are the Ki-67 positive cells. And what they do is they count the ratio from the number of brown to number of blue. And so our algorithm was within the range, and it worked on patches, TMAs, and whole slide images. And it was one of the first of its kind to actually treat this type of nuclei detection problem as a regression problem. And we also tried to focus on by training only using tumor cells. So that's stromal cells, epithelial cells, normal epithelial cells wouldn't get picked up in the output. And so that led us to developing something we called UVNet, because we found actually that there was problems. You can't maybe see it. So this is in distribution data, which always looks amazing. But in out of distribution data, we found that the hematoxylin cells were actually, there was a lot of false negatives and false positives. So we proposed something called UVNet, which is kind of like a UNet architecture, but we incorporated these V blocks that has dense connections to basically preserve the smaller features in our network. And what we found was, if you can see on the left side here, these are the blue cells that were missed. And on the right side with the new architecture was picked up. And as with everything, we always quantify performance. You can see that this is the difference in performance on the negative channel and the difference, and we even got better on the positive channel, on the brown channel. One last work about this is last year, we wanted to improve robustness. We did a similar to the ventricle segmentation. So we generated unsupervised silver standard labels in the target domain using our own algorithm. Then we trained on these silver standard labels from the target. We tuned using a vendor-specific gold standard label. And based on that, we measured performance. And so the bottom row is this proposed model. And you can see F1 in the source domain, it's higher, but most importantly is in the target domain. Look at that improvement, and it's statistically significant as well. And what we can find here is you can see the two feature maps. This is using TSNE, and on the right hand shows the features for the the domain adapted model. And you can see that the features are all mixing for source and target, which indicates that the features that it's learning is similar between the source and target, and that's why we're getting enhanced robustness. This work, I presented it a few weeks ago at Vector in the health session, but it's been a number of years to get to this point. This was kind of the last piece in it. So with the pathologist I work with, she said, here's 500 TMAs, run your algorithm on it, and send us back the results. So we were blinded to what the ground truth results were. And that's a little bit scary because you never know what's going to happen, right? But what we we went against a Perio, Definian's QPath. These are all commercial solutions. And we found that our tool actually provided higher correlation, less error, and better agreement than the commercial tools. And that was recently published in a laboratory investigation. We've worked on nuclei segmentation. We did a gated shape, gated CNN, where you have a regular CNN and you have an edge stream. And this edge stream has gated attention, which basically tries to predict the boundaries. And having this edge loss really helps to segment overlapping objects. Like you can see here, this is with the gated CNN. You can see that the individual nuclei are being separated better, but just using the R2 UNet backbone, you can see that it's not as good. I was going to talk a little bit about color normalization, but I'll just skip past this, but it's a big problem. You can see that if you apply your algorithm at one center, it fails at one place and is good in another. So we wanted to look at why is that, and is color normalization enough? So in the digital pathology community, we just color normalize by default, but does it actually really help performance? And so what we tried to focus on is developing a whole bunch of models using different color normalizations and looking at the metrics. And so what we found was, so here's our unnormalized model we developed. Here's our normalized model. So we measure the DSC on both of the images. If the DSC improves with the color normalization model, we know that color normalization is actually helping it. If the DSC is less, then we know that color normalization is hindering it. And what this graph shows is the unnormalized DSC versus the change in DSC. So any positive DSC means that you're getting an improvement in segmentation performance. And what was interesting is that for images that already had high segmentation, color normalization could improve it or make it worse. But for these really poor performing images, those were the ones that afforded the most improvement. And when we looked at this, we found that we suspect, and we've tried to look at a whole bunch of different theoretical analysis, we suspect that it's related to the contrast in the images. In this one, these are the ones that performed really poorly. You can see like DSC of 0.012 completely missed the cells. When we color normalize it, you can see 0.79, 0.83. So in this case, it's working. But on the other ones, we could see that it actually makes it worse. So we have 0.65 that drops to 0.5, or 0.79 drops to 0.6. And what we suspect is that the contrast in these images is already high enough that when you use color normalization, it actually starts to suppress the contrast. So it might be related to contrast, but we couldn't actually find, we defined a whole bunch of metrics, contrast, color contrast, that it didn't correlate with DSC. We've done mitosis segmentation and detection. This one, we tried segmentation for a very challenging problem. So we're trying to find dividing cells. On in-distribution data, works great. On out-of-distribution data, not so much. So in MyDog 2022, which was a domain generalization challenge, we focused on using a detection network. This is a Cascade RCNN, and in the international competition, our architecture placed 7. We've done renal pathology. The reason why I want to highlight this one is it's very interesting because this space, like I talked about breast cancer, there's a lot of AI in breast cancer. And even Canadian Cancer Society, I have an AI grant, which is kind of new, but at least it's happening there. But for renal pathology, there is like absolutely nothing. It is the poorest published medical specialty in AI. So there we're very resistant to anything that was using computers to analyze imaging data. So it took a while to publish this one, but this is an interesting problem because basically pathologists look at biopsies. They look at the glomeruli, which are the kidney filtration mechanisms. They usually look at the structure of the glomeruli, and then they'll go to a second modality. They'll use light electron microscopy to actually look at the membrane thickness. So we said, what about underserved communities? What about rural, like even Timmins, sometimes they send their slides here. So what about those? Can we eliminate that modality because it's not everywhere and just use AI to predict? And so the first stab we took at it, the pathologist really wanted us to focus on explainable biomarkers. So we did texture, we did size, we did shape, we did color, because they're very new to this space. So we use explainable biomarkers and classical machine learning algorithms to try and differentiate. And we got pretty decent performance. I think it was around 70% performance or 67% performance, which isn't great. So of course, you know, these methods are okay, but they're not the best. So in the next work, we decided to go to try deep learning. Problem with this data set, it was very small, so we had to use pre-training and all that. And to kind of make the pathologist a little bit more accepting of the results, we also did GradCam explainability maps, which tend to focus on the membranous tissue. So even though we thought all these things were necessary, we did another study where we actually we asked pathologists to grade a tissue. This is prostate, and what they do is they provide a Gleason grade, and we gave them with the AI, without the AI, and we also provided them information. So we told them that this decision that we've given you is made by a black box deep learning algorithm. This is made by a transparent algorithm. And funny enough, the provision of this information made no impact on their diagnosis. So the whole black box thing might actually not be that big of an issue. What actually reliance on the AI was actually correlated with the beliefs on AI usefulness. So if they believe that AI was going to be transformative and positive impact on their discipline, then they tend to accept the recommendation of the AI more. In the last couple of years, we've been working on actually integrating our own tools into pathology imaging viewers. Okay, so this is with a local digital pathology company. We're one of their academic partners, and this is a web-based viewer that allows you to view, annotate, and images and also your algorithms. We use their API to basically develop a training and a testing pipeline. And the training pipeline basically pulls annotations, patches the images, and the testing or the inference pipeline patches, does the inferences, recomposes, and uploads the overlays to the imaging viewer. And so this is what one of our tools look like into the viewer. So this is a Ki67 TMA. Again, remember, we're looking at the number of brown cells to the number of blue cells. So these are our predictions here, okay? I could, if there's time later, I can actually open up the viewer, but you see the yellow here, those are the brown detected and the blue detected. So we're now able to start viewing in real time what our algorithm results are. And it's very nice because we can actually just send the link to our collaborators, and they love that, actually. So you can actually, within this viewer, you can embed all types of metadata using the API. You don't only have to do an image overlay, but we're looking at the UI UX and some of the change management of the pathologists, how like they actually appreciate this overlay. And so it's just giving them the answer that I calculated that there's 10% brown. They don't really like that. They like to see the visual results. And what we did is that we were able to then deploy this algorithm in an international pathology study with 90 pathologists. And we published it in scientific reports, and we were able to show that it improves accuracy, agreement, and efficiency of pathologists for Ki-67 assessments. It's one of the largest kind of deployment studies of its kind. And I'll just highlight a few different things. That we had wide demographics from residents all the way to retired pathologists. And we measured, and I don't talk about it today, but accuracy, concordance, and turnaround time with and without the AI. And this is how the study was set up. So they would have to one time score the image without the AI, and another time sometime later with the AI. And we would provide them the AI score as well as, again, the overlay. And it was really kind of funny because when we did this, we actually saw that the pathologist commented on the algorithm missing cells. So on one of the slides, they said, your algorithm missed 3 cells. So like that's kind of, we found that to be kind of interesting. So this was one of the main takeaways from the paper. We showed it makes them faster. We showed it makes them more accurate. But this is one of the big things that I think is really what's going to define the way medicine and AI can really help. So this is the ground truth percentage of the number of brown to number of blue. This is the estimated of the pathologist without the AI. So you can see that there's a consistent underrepresentation of this score, which means that there's a bias, right? So, but with the AI, you can see here that it gets tightened right up, that the ground truth and with aid estimation to the ground truth, there's a very high correlation. What this means is that patients will receive the same treatment irrespective of the pathologist that's reading their scan. And that's really big because if you have a junior pathologist, like every center, they have generalist pathologists and they have specialists. In rural areas, they might not have a specialist pathologist. So if you're, in Timmins or somewhere and your pathology report gets sent to the generalist pathologist, you're going to be assigned the same treatment as if you were being monitored by an expert. And this is really where I believe that AI can start to transform some of this medical imaging practice. I'll just close up. I think that I only have a few slides left. These were their opinions. So agree or strongly agree is the green. So they all took the AI into consideration. They believe it offers appropriate scores. It can improve efficiency and so on, which to me, that means that, or I get the feeling that they trusted the AI, right? They seem to have trusted it. And I don't show it here. We're looking at another trust study, and we found that the AI output is very close to the ground truth. And so that means pathologists trusted it. But what happens if you have an error in your AI tool, right? Are they going to catch it, or are they just going to blindly accept it? So those are kind of some of the things we have to think about. Majority of them thought that, personal implementation or routine implementation is coming. But then look at this. They say personal implementation, not in present condition, not in present condition. I wish we had a follow-up question to say what do they think is the present condition of it, because majority of the people thought that the way it is now, it's not going to work. So, okay, so AI is redesigning medical imaging diagnostics, but are we there yet? So this is the FDA landscape. On the bottom here is just a snapshot of the radiology AI algorithms. There's 671 approved algorithms. On the top is the pathology AI algorithms. There's only one approved. So despite all of this being having FDA approval, like all my pathologists and radiologist colleagues, nobody is using it routinely. So how come? Well, I don't think that we're 100% there yet. There's more work to be done. There's a lot of considerations that we still have to talk about and think about as we are moving towards translation. This was a big one for a long time. How are you going to sell your algorithms? What are the pricing models? In order to go into an organization and convince them to buy your tech, you have to actually show that you have an ROI, a return on investment. So the big one that people were focusing on for a long time was how do you actually make pathologists faster? If they read more cases, they can charge OHIP more. So this was the selling point that they wanted to do, but they haven't been able to show that basically turnaround time improves using digital imaging. And that's probably because the scanning takes forever. So because it's 100,000 by 100,000 micron images, it takes a long time. But there's so many things, deployment, which hospital systems are going to talk to which. It's just getting to DICOM. The pain points and the use cases, I feel like we're starting to get good at that and understand that. But then there's also this trustworthy AI. So there's this continual learning, right? You don't want to get a model that's locked and you go and you implement it at all the hospitals. What happens if you want to change it? Then it's no longer an FDA cleared product. So there's continual learning that's going to have to be put into the regulatory framework. Also IP, you generate these models. Is model IP, or who owns the model if the data comes from the publicly funded healthcare system? So, and that's actually one of the points that's, I believe, making it hard for companies, because like me as an academic, I partner with all our local hospitals. It's wonderful. They're giving me a lot of data. I have more data than I know what to do with. But then when I talk to my industrial partners or collaborators, they always want to sell them data. The hospitals want to sell them data, but it's like a ridiculous price, quite frankly. And, you could buy 100 or whatever cases, but you need big data if you want to make things like foundation models. So there's a lot of work to be done. I couldn't have done any of this work without my team, and this was the only picture I had recently, and I just want to say that in the last 6 months, I've had several students graduate, and I've hired several students again, and we're now more than 50% female, and I'm more than 50%. I've never had that in my entire career, including when I was a student. I was usually always the only woman. And I would also have to thank you to my wonderful clinical collaborators and funders that made this work possible. And I'd love to hear any questions or comments.
Any questions?
Hi, April. Thanks
Hi.
for that presentation.
Thanks.
I'm just wondering, you mentioned that for the digital pathology cases with the radiologists, that you weren't able to get feedback. Do you think that you would try to kind of reapproach that study and do more of like a feedback approach for next time?
Yeah, definitely. Like, you never know what you don't know. So we tried to pick the best questions that we thought would cover the basis. But yeah, like, not in the present condition doesn't really give me any insights on how we could improve it to make them like it more, right? But we have a number of tools that we're getting ready to do something similar with, so we can definitely think about those questions. Yeah, great question. Thanks.
Hi, Professor. I have a question. The cancer is not just an instant result that we need to predict some process in the future. Have you did some work about the prediction process from time to time?
Yeah, like so survival analysis or prognosis?
Yeah, and maybe some normal cells maybe have the bigger possibility to turn to some cancer cells, such like that.
So we haven't done that for cancer cells, because once you cut out the tumor, that's it. you don't usually sample normal tissue for like cancer diagnostics, but in neuroimaging, we have actually stripped away all the lesions, all the kind of pathology, and looked at the normal area. And we actually found that there is early signs of disease there. And that's super important because you're saying that this person is already on the trajectory to a stroke or dementia. We are starting to work on more survival models now that we have kind of complete biomarker systems. We're not the type of lab that just extracts all the features and hopes something comes out of it. We're kind of carefully crafting everything to be mimicking what the radiologist and pathologist does. And so we kind of have gotten there with the frameworks to start to do some of this survival and prediction, like prognosis, basically. You know, given this image, we think that you have a 50% chance of having a stroke, or, you know, you might pass away from the cancer. So that is the future. Prognostication is really the future. I feel like it goes from quantification to like monitoring to stratification, and then this prognostication, which is really the future. And some of those multimodal models that we're starting to look at is going to really play a huge role there, because you know, one piece of information, even though imaging is very strong and one of the strongest in pipelines, you know, disease is represented in multiple dimensions. So multimodal models, I think, will be key.
Couple questions online.
Yep.
Just to speak in here. From Caroline Chong, what do you think clinicians need to have confidence and trust using AI models long-term in their practice?
You know, some of it is this explainability piece, because I've been working with pathologists for 10 years and I've seen their kind of change management, that initially digitization was a big deal, but now they seem to be accepting it actually. And if you go to any of the pathology visions, pathology informatics, the whole thing is AI for digital pathology. So I think, you know, to get them to accept it, they have to use it and they have to understand it. And part of the way that we try to bridge that gap is that we do development with pathologists for pathologists. So we don't try to design a solution that's going to fit into. So having pathologists involved really, you know, makes it's almost like product management, in that you are basically ensuring that you are answering a question from a pathologist. So if you come to them and say, I have some fancy dancy model that can automatically classify, but then you don't give them any information about what it does and how it does it, you know, they're less likely to be inclined to trust it. So there has to be some explainability. They should be involved in the process of development and deployment, and you know, come with us on the journey of translation.
We have a couple more online. So from Arbor, great talk. Going back to the brain health neuroimaging works, have you looked at integrating and correlating behavioral features like neuropsych, eye tracking, audiology with imaging data biomarkers?
So we haven't created multimodal models that combine it, but we have correlated to neuropsych cognitive scores like MoCA, MMSE. We even looked at delusions and psychosis. We started looking at depression to look at subtle features in the brain. So one of the things that I am interested in, now that we've grown our database significantly as well, is maybe incorporating genomics information, right? Like the clinical variables, like to create a model, you know, what would you use? Like a vision transformer that embeds both the text but it's one single word. It's like, okay, or one single value. So I don't think creating a multimodal model from those would be so useful compared to like genomics, phenomics, or some other multi-dimensional data set.
Thank you. One last question from online. Hi, April, from Michael. Thanks so much for your talk. Referring to your pre-processing tools, I'm aware of other popular neuroimaging packages like ANTS and FreeSurfer. Have you done any head-to-head comparison between your lab's in-house tools and these packages?
Yes.
Yes. Okay.
That's what we always do. Yeah. So I always tell my students, if there's something that solves the problem that you're trying to do, then why not just use it? So if it works, so we always try what's existing first. If it doesn't work, we understand the failure modes and then we try to improve upon it. So always, yes, we compare to what's out there first.
Wonderful, thank you. And I think we've got 1 minute left if there's another question in the room. Fabulous, there we go.
I have one last question. Earlier in your presentation, you talked about the prediction for conversion to Alzheimer's disease.
Mhm.
I'm curious if this is like widespread, like used widespread, and what would help physicians or organizations use this more often, especially with the increase in kind of medications that can slow the progression.
Yeah, so that's an excellent question. In the last few years, lecanemab has come out and it basically is an amyloid beta clearing drug. So that's kind of where we're going with some of these. Can we use these biomarker frameworks to identify candidates? You know, but if somebody isn't going to convert to Alzheimer's disease, they probably won't have amyloid beta accumulation. So they probably don't need to actually get that drug. And there's like a huge a risk of ARIA, so which is edema and hemorrhage from these drugs. And so you don't want to give somebody who is not going to convert to Alzheimer's these drugs. So this is exactly where we're going. The funny thing is that clinicians all want these metrics, but they're a little bit uncertain about how they would incorporate into their clinical practice. So that's kind of what we're looking at in the next year or so, is talking to a lot of physicians, doing some UI, UX, understand how this could change reporting, and to do like a clinical trial around that. Yeah, great question.
Right. Let's thank our speaker again.